In this video, we will discuss about primary brain tumor, that is GBM, how they induce the immune suppression. So before this finding, it has been very difficult to treat the patients that are affected by this GBM or primary brain tumor, and it has been find out that if this primary brain tumor GBM is removed from the patient, there is a high possibility of reoccurrence of this GBM within the patient, and the survival rate for the this kind of patients is very low. And one of the reason is because of the immune suppression that is caused by GBM. However, after this finding, there is a possibility that this immune suppression uh, can be reduced to a greater extent by blocking out the receptors that are present on the macrophages, interact with the GBM and cause immune suppression. One of the receptors that are popularly known as checkpoint inhibitors, that is PD-1 receptors that interact with the PDL1 that are present on the GBM. However, in a study that has been conducted on the animal mouse models, it has been found that even though they suppress this PD-1 or they block these PD-1 receptors, the immune suppression is not reduced greatly. However, in this finding, it has been found out that the CLEC1 receptors that are present on the macrophages also been interact with the sialic acids that are overexpressed on these primary brain tumors, and that leads to the recruitment of the T regulatory cells. And this T regulatory cells has a function to suppress the immune cells. So it will also deactivate the T cells. So if so, in this study, what they have done is they not just block the PD-1 receptors, but also they block the cyclin receptors that are present on the macrophages. And by blocking both of these receptors, they can see that the amount of activated T cells are more within the animal model, and and this might leads to a possibility that by blocking out these two receptors, there is a high probability of reducing the immune suppression in the patients. So how this macrophages in general term actually work, it will convert it into this tumor associated macrophages 2. After drug treatment, it can convert it into this tumor associated macrophages 1, and then it will affect the tumor cells and trigger apoptosis when these receptors are being blocked. And when the receptors are not being blocked, what happens is that these sialic acids that are overexpressed on the GBM might interact with the selectin receptors, and that might lead to the recruitment of the T regulatory cells. At the same time, this what happens in terms of this general checkpoint inhibitor therapy is that B cells will take away the antigen of this GBM, and that pass on to the dendritic cells, and thereby the dendritic cells will recruit this T cells or will generate antibodies against the GBM. However, the activated T cells will get suppressed because of the interaction of this PDL1 antigens to these PD1 receptors. So by blocking these two receptors, so these anti-PD1 receptors and this anti-CD47 antibodies, uh, there could be a possibility that patients that are affected with brain tumor might not cause high immunosuppression, and they might be able to increase the immune response against this primary brain tumor. And the way by which this anti-PD1 and anti-CD47 can be introduced in the patient is one of the examples that have been one of the possibilities that is possible at this point of time is using CAR T cell therapy, where they will transduce they will take the T cells from the patient and modify them using the anti-PD1 receptors and anti-CD47 receptors. Thereby, they will be able to block these anti-PD1 receptors on the T cells. At the same time, this anti-CD47 receptors will be able to block the CLEC9 receptors on CLEC9 receptors on or CLEC9 antigens on this GBM, and thereby that might prevent the intercellular triggering that will lead to the recruitment of these T regulatory cells, and thus the immune suppression will be reduced, and the immune efficiency against this GBM will be increased, and the possibility for the reoccurrence of this primary brain tumor might be reduced greatly within the patients, compared to the case where only this anti-P1, where only this PD1 receptors is blocked. So I hope you got an idea how we can uh, improve the conditions of the patients that are affected with the primary brain tumor, that is GBM, and reduce the immune suppression that is caused by this GBM. If you need to understand more about this, how this primary brain tumor leads to the immune suppression and how it can be overcome, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.